This work is done in China, new place. I'm going to join next month. Current situation is in UCLA and Creighton University in Omaha, Nebraska. But this is through the international collaboration. I'm in charge of the School of Pharmaceutical Science and Technology. Donnie is a beautiful, charming place to stay. That's why I'm joining them. So today I'm talking about the microRNA regulation in cancer stem cells. As you know, the previous several talks is heavily focused on the therapeutic agents development. Now I'm going to use the cancer stem cell as a target to develop a new agent. I call it microRNA, hopefully. I want to quickly through the cancer statistics. Everybody, as you know, breast cancer still is the leading death of cancer in women in the world, even in the United States. Just a skip it. Why it is still the highest? It's getting better, but what the reason was generated the high incidence rate? And so because I know the early diagnosis is a big problem because we lack of effective approaches to diagnose it earlier. But most important question is based on recent research, the recurrence and the relapse of the therapy, which is a major reason initiated and maintained by remaining cancer tissue stem cells from either residual tumors or those with the intrinsic resistance to adjuvant therapy. So now that's why we focus on the use of the cancer stem cells as a therapeutic target. Now, specific focus on the breast cancer stem cells. As you know, the cancer stem cells come from the stem cells. What's the exactly etiology? Still unknown and not clear. It's going to come from the regular stem cell because it's during the process differentiation and mitosis. So there's a number of factors could infect the process mitosis, cause a number of the mutation. So this is I don't want to spend too much time on that. So we are interested in the two types of cells. As I said, CD44 and CD24 is two cell surface markers expressed in these culture cells. And we are going to use that as a tool, as a target to start in the microRNA regulation. Quick through that. That's the biosynthesis of the microRNA initially through the pre-microRNA processed by the enzyme called DROSA and translocated to the cytosol. Further processed through a number of the enzymes, especially dicer, and becomes a mature microRNA, perform the functionality. Through the previous study, I was done in Creighton and collaborated with UCLA Pancreatic Cancer Center, but pancreatic cancer is still working on breast cancer as well. We are collecting tissues from patient, and this is some of the patient tissues from the Department of Pathology, UCLA, and some of them from the Creighton Pathology. We are looking for focus on 3 stages: benign tissue and DCIS and invasive cases. We found out through the RT-PCR platform, which we modified. We identified the number of microRNA, which is upregulated, downregulated. But most importantly, we think we're interested in the microRNA 34A. Of course, the number of other microRNAs, because the mechanism why this microRNA downregulated in the cancer case, not clear yet. Most of this speculation because of the promoter of the microRNA is highly methylated, epigenetically regulated. So this study, we just to give us a heads up to see what the underlying mechanism of those microRNA play in the cancer status. As you know, so hopefully, as I say, use that as a target to see if microRNA 34A can be developed as the therapeutic agents. Because this is especially for the cancer stem cells. So we are starting from the using the breast cancer cell line, which is a mobilized cell line, is a MCF7. As I say, MCF7 can express cell phenotype is CD44 and CD24. So we're interested in CD44 positive and CD24 low expressed phenotype. We use that as a target, and we see if it's microRNA 34 can be regulate the process. Interesting, first of all, we look at the hypothesis whether the SERT1, okay, so SERT1 is not NAD-dependent enzyme. It's an amylase enzyme. This is, has been demonstrated as a target of microRNA 34A in other cancers, regular cancer. But why we are still interested in that? Because there's no one is work on the breast cancer stem cell. We don't know what the role of the this axis of microRNA 34A and SERT1 play in the initiate or regulate the breast cancer stem cell process. So we first look at the endogenous level to see. As you can see from the first slide, it shows this is a parental cell line. It's MCF7. This is we separate use the markers. And non-specific, as you can see, is microRNA 34 is significantly downregulated. And at the same time, we can see the SERT1, which we call the target, is operated. This definitely is a reciprocal relationship. And protein as well. You can see the protein level upregulating the breast cancer stem cells, and that is the chart plot. So that means demonstrate the reciprocal relationship between microRNA 34A and SERT1. And now if we can manipulate the microRNA 34 and SERT1 axis, see how this manipulate can affect the cell growth, stemness, self-renewal. So we are running this experiment by either downregulate the SERT1 using the silent RNA, shRNA of SERT1, or we epitopic expression of microRNA 34 in the cells. As you can see, we're using the different, this is silence the genes. You can see it's efficient, sufficient, suppress the expression of SERT1. The 34, if we, the 34, same thing if we, first, this is a demonstrate the efficiency of the expression, of epitopic expression. And for instance, if the cell line overexpress 34A, we can see significantly suppress the SERT1. We will see the cell line, the endogenous level of this breast cancer stem cells, as you can see, the highly expressed in the, as we demonstrated previously. Now we will hypothesize if we, by alteration of this axis, whether the alteration of the axis can affect the growth potential. We are using the cell growth assay, it's a colony counting, cell counting assay, and we demonstrate it is either overexpressed microRNA 34A or silent the gene, SERT1 gene, significantly inhibits cell growth. What about on the other hand, we also run through the colony assay, colony formation. As you can see, 35A overexpressed cell treatment is significantly suppressed, demonstrated by the by the chart or not done there. And if we silence the SOL1 gene, it will show the similar pattern with significant, the color information is significantly inhibited. Now we want to see the cell behaviors, proliferation. We're using the dual staining PE versus the Physi. As you can see, the MYR38 overexpressed cell is generated pre-apoptotic cell death population, significantly causes cell apoptosis. What about the mammosphere formation? This is the mammosphere formation capacity is another features of the cancer stem cell of the self-renewal. As you can see, the number of mammosphere formation significantly decreased, either upregulated male microRNA 34A or downregulated silence the gene SOL1. Now, we are concerned with standard breast cancer stem cells. What the biomarkers and cell surface demonstrated to see response behavior looks like. So we are looking at the LDH1, NANAC, and the BMI1. And those genes is consistently downregulated, treated with, in the cells treated with either microRNA 34A or silenced the genes, silence as SOL1 genes. This is the debauch just demonstrate the significant difference. And now the question whether alteration of the this axis can suppress the important biomarker of the stem cells, NANAC. It is. This is the Debye staining, and which significantly shows the suppress the breast cancer stem cells versus compared to the control cells. And if we silence the gene, show similar pattern as we demonstrated that by upregulated, by epitopic expressed the microRNA 34A. Now, the modulation of this axis can enhance MSF cells apoptosis. This is a further demonstrate the relationship. I will just click through, and you will see the cells compared to the control. This is a control group. This is overexpressed 34A. This is a silence the SOL1. They significantly inhibit. Okay, thank you. And now we want to say this is the previous experiment we done in vitro. Now what happened to behavior in the tumor in vivo? So we are using the new animal mass to cells eventually transfected using the antivirus and Stepsilna and inject to the fat pad to see how the cells, the microRNA, those transfected cells behavior in vivo. As you can see, it shows a similar pattern as we demonstrated in vitro. Either upregulated microRNA 34A or silence the genes of SOL1, significantly inhibits tumor growth. This is the tumor size, as you can see, collected from the tumor size is significantly inhibited by either upregulated 34A or downregulated SOL1 genes. Clearly, it shows the pathologic slice HE stain and shows the tumor density, and SOL1 gene. This is a treatment. This is a control. Is a significant suppressive expression of the gene in the tissue. RT-PCR further confirmed the xenograph, the expression of 34A suppress the upregulating the tissue, which is transfected, antivirus transfected. Protein level, it shows similar thing, is confirmed what we discovered in vitro. This is a kind of a tentative mechanism we propose, because as we know, transcription factor P53 is upregulated microRNA 34A. 34A has a number, microRNA has numbers of targets, can target ESO1, which is we are demonstrating on the other groups as well, as a direct target ESO1, and also target CD44 and Notch. This is a proof. Notch signaling is just published recently, cancer research by Dr. Song in China. Eventually target the breast cancer stem cells, but the mechanism in the downstream effector we still don't unknown. The mass of the study is undertaken. So I'm going to end my talk right here. This is my the research, special research through international collaboration before John's group and Chinese government. No, the university has we have won the grants. We went through the research. This is my fellow over here, United States, in creating a UCLA, part of a group. Thank you very much for your attention.